Thank you, and thanks to the Alliance for the opportunity to introduce Regencor to you all today. I don't know what happened to the R. The ethers seem to have misplaced it. So as our, the green button. Come on, Tom. So as our name implies, our mission is to regenerate the myocardium after infarction. And the founding technology was published in Nature at the end of last year. This is not working. So this is an illustration from the New England Journal editorial that commented on the Nature publication early this year, and it's the elevator pitch. So the story is that if we apply to the infarcted ventricle a collagen patch containing human recombinant FSTL1, follistatin-like 1, that's produced in bacteria, that's clearly important, we get striking regeneration of cardiac structure. So increased numbers of dividing cardiomyocytes, reduced scarring, and increased vessel formation. And this structural regeneration leads to a restoration of function, and in the mice, improved survival. Next, please. So we'll go through most of these points during the talk. We're a new company spun out of Stanford and the Burnham. We have a pretty experienced management team and board of consultants. We have a large unmet medical need in ischemic cardiomyopathy. We have proof of concept published in Nature in 2 animal species. We have pretty broad composition of matter patent applications in process, one co-owned by the company. And in collaboration with our consultants and advisors, we've put together our product development plan. We're developing a delivery cannula. We have a large animal preclinical partner in the University Medical Center at Utrecht. And I'll spend a second on the last point. The Nature paper sparked a lot of interest from large pharma, and we've spent most of the past year under NDAs talking to 4 of them. And we now have an executed letter of intent with 1 for a fully funded program through phase 1. So if this closes, it's obviously a great source of non-dilutive financing for the company, and after closure, we would probably go on for a traditional Series A. Next, please. This is the management team. So the founders at the top are Pilar Ruiz Lozano and her husband, Mark Mercola. They are outstanding scientists in cardiac regenerative biology. Al Chin, who's actually my neighbor, you can't make that up, in Palo Alto, is the developer of the injection cannula that we'll speak to. Roger Hajar is the head of cardiovascular medicine at Mount Sinai and is our in-house cardiologist. Gladys Monroy, whom I've worked with for many years, was former co-chair of IP at Morrison & Forrester. And I've also brought on my partner at Geron Corporation, my CFO for 12 years, David Greenwood, and Jane Lepkowski, who's been my scientific partner for over 30 years, who needs no introduction to this group. Next, please. So we have pretty deep IP. We have executed the deal with the Burnham, and we are about to execute the deal with Stanford. And in addition, we will license 2 issued patents from BU. And I should say that the Burnham patent has received a very clean international search report and written opinion by the U.S. Patent and Trademark Office, which augurs well for the international phase of prosecution. Next, please. So the market is big, it's a bad disease, and there's no cure. The link to this opportunity in ischemic cardiomyopathy and our technology is that the cells, half these patients have so-called hibernating myocardium, which are dedifferentiated cardiomyocytes that have a cellular phenotype very close to the cells in the zebrafish model, which I'll talk about, that regenerate, and they're also similar to the cells we identified by drug-induced Krelok's sulfate mapping to be responsive to this protein in the mouse model. Next. Next. You're going to have to give me an extra 5 minutes. Go. Next. So the paper at the bottom received quite a bit of attention. It was nominated by Faculty 1000 Prime. It's been downloaded over 60,000 times, and it's in the upper 75 percentile of all Nature publications. Next. So the star of the show is the epicardium, which has been very heavily implicated in regeneration in lower vertebrates who can regenerate their hearts after injury. And in fact, mutations to the epicardium in those species leads to a failure to regenerate the epicardium. So does this one cell layer thick layer have any impact on human regeneration? Next. So the initial observation was if you culture dividing cardiomyocytes with epicardial cell lines, you get profound proliferation in vitro. And that's repeated with the conditioned media from the epicardial cell line. Next. So that led to an extensive screening program, actually here at the Burnham, where we found over 300 single reads in a liquid chromatography mass spec screening program. We purified over 100 of them and tested them one at a time in that assay. And at the end of the day, there's only one factor that is responsible for the proliferation, and that's a particular form of follistatin-like 1 protein. Next. Next. So here's a story in zebrafish. You can actually snip off the tip of their ventricle, and they will scarlessly regenerate in a matter of weeks. And on the right side, you see an in situ hybridization showing that the zebrafish has two genes for follistatin, A and B. And A is fully glycosylated, and that is not activated during regeneration, top line, as opposed to 1B, which is hypoglycosylated, and that's abundantly expressed in the epicardium after injury. Next. In contrast, mammals that do not regenerate do produce FSTL1 in the epicardium in a resting state, but after injury, the epicardial production of the protein disappears. Next, where does it go? Complicated slide. The bottom line is, after injury in the mammals, the production of the protein is taken up by the myocardium, exuberantly so, yet it does not regenerate. So what's going on? Bottom line here, which I don't have time to go through in detail, is that the epicardial FSTL1 is hypoglycosylated. The myocardial form is heavily glycosylated. And fortunately, to work this out, there are available in the research marketplace human recombinant FSTL1 made in bacteria and in mammalian cells. And as you remember, bacterial cells cannot glycosylate. And in the lower right-hand portion, you'll see that the only factor that is active in vitro in getting the cardiomyocytes to proliferate is the non-glycosylated bacterial form. The glycosylated form is no different than PBS. Next. Next. So, the patch. It's highly bioengineered. After polymerization, we put about 10 micrograms per patch of the protein in it. It's then compressed to a very narrow spec. It basically has the elastic modulus of embryonic myocardium, and that was deliberately designed to support the contractile apparatus of regenerating cardiomyocytes. So the stiffness of this is about 5 to 10 kilopascals. To put that into perspective, adult myocardium, that's normal, is about 30 to 40. The scar after an infarction is about 100. So this is very soft collagen. Next. Here's the animal studies from the Nature paper. So on the left is fractional shortening as deduced by 2D echocardiography. The blue line on the top are the sham controls, which don't decline significantly. The red at the bottom on your left are the MI only that has profound reduction in fractional shortening. The green is the MI plus patch plus the protein, which at the end of 3 months is indistinguishable in terms of fractional shortening to the sham controls. There's some non-significant impact of patch alone that's not due, that is due to decreased wall thinning and support. But the histologic evidence of myocardial proliferation and blood vessel formation shown on the right is not associated with patch alone. It only takes place in patch plus protein. And as you can see on the right, there's a significant survival advantage conferred on these mice with patch plus protein, which is actually not significantly different from the sham controls. Next. We've moved on to pigs, large pigs, Yorkshire pigs. And here, using DEMRI, delayed enhancement MRIs, we can show significant improvements in ejection fraction. Here, the patch is applied a week after infarction, and you can see you go from 51% pre-infarction down to a low of 32 and back up to 40 after about a month. Next. We can also look at wall motion, and you see the same effect in large pigs, and also in red is an important attribute of significant scar reduction. So the blue is the scar area right after infarction, and you can see it significantly reduced 33 days later. Next. So, product development. Next. This is the top-line product development work plan that we've agreed to with our potential pharmaceutical partner. Essentially, it's scaling up to 6 large groups of large pigs to confirm and extend the mouse work. We're going to look at very different ways of delivering this. We like the patch because it's an independent way to deliver the protein regardless of the status of the coronary arteries. But we will look at injection, we'll look at gene injections, we'll look at encapsulation. And if we survive the stage A, we will move on to a typical CMC preclinical package leading to the phase 1 first-in-man study. Next, please. A little bit about the cannula. There is an existence now under Al Chin's development, an endoscopic epicardial catheter that is being used today in large pigs to do gene therapy injections epicardially. And so what he's doing for us is replacing on the bottom the injection port with a dual-pronged cannula that actually will apply under direct visualization the patch on the epicardium and attach it using surgical cement. Next. The approach, we think, will be an outpatient approach, transcostally. So the catheter is injected superior to the pleural cavity, so you don't need intubation. It enters the pericardial space, again under direct visualization, and the patch is applied. Next slide shows, on the left, the actual visualization of the myocardium, of the infarct through the cannula, and on the right is the actual same animal visualized directly. So you have very good control for placement of the patch. Next. We've enjoyed significant support from our academic partners at Utrecht, who have considered the device to be a breakthrough, and they've actually dedicated matching funds for a lot of the large animal preclinical work, including the first-in-man clinical study. Next. So you've heard most of this. We're a new company spun out of Stanford and the Sanford Burnham. We have some pretty experienced people helping us develop the plan. There's a very large unmet medical need that we think mechanistically we can penetrate a portion of. The Nature paper was extremely well received. We have pretty good patent coverage for the technology. Once we commercialize, if we commercialize with the cannula, the company will have 2 revenue sources, the protein patch and the cannula, which we own without a royalty burden. We've gone pretty far along in terms of CMC plan, first-in-man clinical protocol, GMP manufacturing, and helped by the fact that bacterially produced material, which is active, is already available for the research marketplace. So our manufacturing consultants really see no headwinds in terms of producing the protein. And again, a lot of the large animal work will be done at Utrecht, and hopefully before the end of this year, we will close on a significant funded product development collaboration with a large pharmaceutical firm that will provide a significant amount of non-dilutive financing, after which we will probably do a Series A. Thanks very much, and I'm sorry for the slides.